Hi everyone, I'm Philippe Garabedian, the Director of Operation of Myriad, and I'm here to present you the Videodrop, the technology we have developed to see and count the nanoparticles. What is the context? Gene and cell therapy are booming, and the need for cost control and reliability of bioproduct call for a new technological response. In this context, we have identified a need, which is a solution to be used in process, in real time, and on viruses production line, to allow the measure of the concentration and the size of nanoparticles, like viruses, that allow to see and detect big particles, which mean aggregates potentially present in those solutions of nanoparticles, like lentiviral vector solution. To, in the end, continuously monitor the production of these viral vectors, because they are too small to be detected with a standard microscope. To validate this need, we have built a collaboration with Aratinga, a French biotech company, now part of Rex Généraux. Aratinga is developing a CAR T-cell treatment. You can see on the diagram, on the left, all the steps of their bioproduction of viruses. It is, as you know, a complex process. It lasts 4 weeks. One batch costs at least 500,000 euros, and their scientists are almost blind until the final quality control. During our 12 months of collaboration, we have identified with them where we should ideally place the Videodrop during the bioproduction. We identified 7 places in the upstream and in the downstream process, and even one when the therapy arrives at the hospital and before the injection to the patient. It clearly justified our vision. There is a need for a tool for in-process controls. Now let me present you Myriad. Myriad was born in 2017 from a partnership of Quattrocento, a company builder in Paris, the foundation Paris Sciences et Lettres, which is a research organization in the center of Paris, and the scientific team. Today, Myriad imaging technology is currently protected by a family of 2 patents. But before I explain you how the technology works, I will tell you where it comes from, and precisely what is the Tara Expedition Ocean. Tara Expedition Ocean is a boat that traveled the ocean. It was funded by France, Germany, and Great Britain. It has the objective to study the impact of global warming and climate change on the planktonic system and corals. Martine Bocara, who is a virologist at the Université Pierre et Marie Curie in Paris, was a scientist on the project, and she wanted to know if the global warming had an impact on the viruses population. Are there more or are there less viruses? She wanted, in the end, to count those viruses. And standard techniques were not answering her needs. They were too time-consuming or complex to be used on the hundreds of samples she has to analyze. So she was looking for a fast and robust method for counting viruses in those seawater samples. So, to study them, she asked her brother, Claude, to build her a system. Claude is a specialist in optics at the London Institute in Paris, and is a physicist specialized in interferometry. He therefore developed for her a special interferometer. The Videodrop was born. Now let me present you a short video. So the analysis with the Videodrop of the size and concentration of nanoparticles will be done in almost 40 seconds, from the beginning to the end of the process. On the left, you have the software interface. On the right, you have the system. The user will take a sample of 5 to 10 microliters. Using a pipette, he will place them under the objective of the Videodrop. The user will lock the system and launch the acquisition. It lasts 6 seconds. 1, 2, 3, 4, 5, 6. And you get the image, which is the representation of the interferences generated by the nanoparticles. Then you will get the resultant concentration in particles per milliliter and the modular diameter. So after watching the demo, let me give you some more details regarding the technology. Scientists need to measure nanometric objects. We can count objects such as viruses because we measure size from 30 nanometers to 10 microns. We do it in a concentration range from 10 to the power 8 to 10 to the power 11 particles per milliliter. Scientific work on expensive samples. We only use a single drop of 5 to 10 microliters. Scientists need to save time. We measure the nanoparticles concentration and size in 40 seconds, almost in real time, and it's easy to use. And scientists need to work on raw samples with no purification and no preparation. So we think these features make the Videodrop the ideal tool for in-process controls in bioproduction. Going deeper into the technology, it is interesting to know that the innovative idea of the scientific team is to have transformed a standard microscope into an interferometer. On the same image, you can get a nanoscale detection mode and a microscopy image. So it is possible to count nanoparticles and identify aggregates with the same device. Currently, we have 3 publications demonstrating the power of the technology. Now I will tell you more about the application. So what can we see with the Videodrop? We focus on viruses, but we have been approached by people working on phages and extracellular vesicles. For viruses, we can continuously monitor viral vector solutions like lentiviruses or adenoviruses that are used in gene and cell therapy. For phages, we can analyze complex mixtures of phages T4, lambda for application in phagotherapy. And for extracellular vesicles, we can distinguish, count the different types of extracellular vesicles that are used to vectorize, diagnose, or treat. Now let me show you some examples of what can be seen effectively. So Videodrop sees in real time a huge diversity of objects. In square 1, you have the example of EVs in the blood plasma and with some aggregates. In the square 2, we have the image of HIV viruses. The HIV viruses are circled in orange, and in green you can see their displacements. The displacement is used to measure the size. In the square 3, you can see low concentrated plastic nanobits at 100 nanometers. It's typically the solution we use for our development in R&D. And in square 4, you have the image of lentiviruses. The lentiviruses are those black or white spots all over the image. Now I will present you the interface of the system. In the center, we have the image where you can see the actual interferences generated by the nanoparticles. On the right, you will have a diagram showing the dispersion of the measure of each nanoparticles that are present in the sample. Then you will have the concentration in particles per milliliter and the modular diameter of particles present into the solution. So in the end, the technology allowed the visualization of nanoparticles and macroscopic objects, the measurement of concentration and size of nanoparticles. We do it in a single drop. The results are obtained in 40 seconds. It's easy to use. We don't need any label, and we do it with almost no purification. These measures are done in a range from 30 nanometer to 10 micron, and in concentration from 10 to the power 8 to 10 to the power 11 particles per milliliter. Thank you very much for your attention, and I can now answer your questions. Thank you very much.